In this video, we will address the parasite life cycle and its transmission. You will also learn about pathogenesis and immunity. Remember, both cutaneous and visceral leishmaniasis are caused by a protozoan parasite of the genus Leishmania. This genus was named Leishmania in honor of the Scottish pathologist William Boog Leishman, who first described the protozoa in 1900. We distinguish 30 species of Leishmania worldwide. Visceral leishmaniasis is caused by Leishmania donovani and Leishmania infantum in the Old World. Leishmania infantum occurs mainly around the Mediterranean basin. In East Africa, it is Leishmania donovani that is responsible for visceral leishmaniasis. The Leishmania parasite, regardless of the resulting form of leishmaniasis, needs two hosts to complete its life cycle. The host that acts as a reservoir of the parasite is a vertebrate, human or animal. The second host is called an intermediate host and allows transmission of the parasite. Here it is a blood-sucking sandfly. The parasite has two developmental stages, depending on the host involved. The amastigot stage is found in the vertebrate host. It is an intracellular stage with an ovoid form, 2 to 4 micrometers in size. It is mainly found inside macrophages, where they multiply by binary fission. It is transmitted to the vector through the blood. Once inside the sandfly, the parasite becomes a promastigot. At this stage, it is extracellular and has a flagellated form. Its size is around 15 to 20 micrometers long and 1 to 2 micrometers wide. It is the infective form for vertebrates. Let's have a closer look at the Leishmania protozoa life cycle. A blood-sucking sandfly will feed on the blood of a human or other vertebrate that is infected with a parasite. Together with the blood, the sandfly will ingest amastigots. Once in the midgut of the sandfly, the amastigot will become promastigot and will multiply inside the sandfly. During several days, the promastigots will accumulate in the insect's proboscis, which is the mouth part used to pierce the skin. The sandfly is now infectious. While blood feeding on a new human, the promastigots will enter the human organism at the bite site. They will then be phagocytized by macrophages. In the macrophages, they will become amastigots and will multiply and spread in the human body through more mononuclear phagocytic cells. The bitten human is now a reservoir of the parasite. Transmission of visceral leishmaniasis in East Africa and Asia is anthropootic. This means that infection goes from human to human, except in Sudan, where transmission from animal to human has been observed. The vector of the parasite in East Africa and Asia is a sandfly of the genus Phlebotomus. Out of the 500 known species of phlebotomine sandflies, less than 10% of them have been identified as Leishmania vectors. In areas where the disease is endemic, it is estimated that less than 1% of the sandfly population is infected. Also, only female sandflies may act as vector, as they're the only ones that suck blood during their reproductive process. In East Africa and Asia, visceral leishmaniasis is caused by the protozoa Leishmania donovani, which may be transmitted to humans by different phlebotomine sandfly species. Phlebotomus orientalis is found in Sudan and North Ethiopia. In the southern areas, such as South Ethiopia, Kenya, and Uganda, two different species act as a vector of the disease. These are Phlebotomus martini and Phlebotomus celiae. Finally, in Asia, the parasite is transmitted by Phlebotomus argentines. Remember, in Module 1, we learned that phlebotomine sandflies responsible for transmission of Leishmania donovani are found in quite diverse habitats. On the one hand, they are found in natural settings where moist, warm, and organic matter are available for their larvae to develop. On the other hand, open sewage, and household rubbish provide also optimal breeding sites. The size of phlebotomine sandflies ranges between 2 and 3 millimeters, which is much smaller than that of a mosquito. Due to their size, they are poor flyers with a short, hopping flight style. Therefore, they are poor dispersers and are more often found in a radius of a few hundred meters from their breeding site. People living close to breeding sites are thus at risk of being bitten and infected with Leishmania parasites. Given their small size, sandflies can pass through routinely used mosquito nets. Only impregnated nets can limit exposure to the sandfly because they become intoxicated with the insecticide while passing through the mesh. However, bites occur more often outside dwellings. Sandflies cannot bite through clothing, therefore they bite exposed parts of the body, usually the face and hands. The Leishmania promastigot enters the human skin through the wound caused by the blood-feeding sandfly. This intrusion triggers an immune response to fight against the parasite. Once phagocytized, the parasite is able to inhibit production of nitric oxide that would kill it inside the macrophage. The phagocytized parasite then becomes an amastigot and multiplies inside the cell until it ruptures. This releases plenty of amastigots that infect other phagocytic cells. Such an intracellular infection requires a strong Th1 immune response to get rid of the parasite. The immune response may be influenced by other substances injected through the saliva of the sandfly. Among these products are a gel produced by the Leishmania parasite and some saliva proteins. Both substances appear to influence the immune response that would shift towards a Th2 response. While subclinical infections resolve spontaneously through an efficient immune response, up to 25% of infected people fail to produce an appropriate immune response, allowing expansion of the parasite in the organism. The parasite's progression hinders the functioning of the infected organ and weakens the immune system, leading to many health problems. Clinical symptoms of visceral leishmaniasis are presented in the next module. Infected patients who develop clinical visceral leishmaniasis for the first time in their lives are called primary cases. Disease left untreated is fatal in about 95% of these cases. Anti-leishmanial treatment is necessary to allow the immune system to respond with an appropriate cellular immune response. Cured patients will be immunized for life against the infecting strain. Some patients may suffer from a return of the disease after a period of improvement of several months. These cases are referred to as relapses. The rate of relapse is low in immunocompetent patients, but is very frequent in those who are immune compromised.